Good evening, ladies and gentlemen, and good morning, special to Dr. Anna Meiji, because now it's 10 a.m. in German, I guess. Welcome to guest lecturing under 3-in-1 program of fertilizer and its application technology. It's being a great honor for me to welcome you here. Before we go to the program, I would like to express our sincere gratitude to the Excellencies Head of Soil Science Department of Brawijaya University, Dr. Syarul Kurniawan, and in this case, he also will be lead the discussion today. And all of the lectures of Agriculture Faculty, especially Soil Science Department, and the special our guest lecturer from George August Universität Göttingen, Germany, and all of the audience who follow this lecturing both from Zoom and YouTube. We have many audiences from bachelor degree undergraduate program and also from master program. Well, ladies and gentlemen, today we have a great opportunity to learn about greenhouse gas emission from different agricultural systems that will be presented by Dr. Anna Meiji from George August Universität Göttingen, Germany. Then, before we start the program, I would like to greet Dr. Anna Meiji because it is our first meeting. How do you do, ma'am?
Thank you very much for the introduction. Yeah, nice to meet you. And it's a pleasure for me to be here.
It's my pleasure. Thank you for your coming. Well, we are now in our main agenda, namely presentation and discussion. I do hope Dr. Syarul Kurniawan could lead the discussion because I guess Dr. Syarul is your best friend. To Dr. Syarul Kurniawan, time is yours.
Okay, thank you very much, Bu Nova. So, first of all, I would like to thank first the Honorable to Dr. Anna Meiji from the Agronomy Department, Faculty of Agriculture, University George August Universität of Göttingen. And then the honorable of lecturer from soil department and as well as from the other department in the Faculty of Agriculture, University of Brawijaya, and all students, including agro-ecotechnology and the postgraduate program of soil and water management. First of all, let us praise and thank the presence of Allah Almighty for the abundance of grace and the joy of all of us can still gather in the good health. Second, on the behalf of Soil Science Department, I would like to thanks to Dr. Anna Meiji that's for the willingness to be the international guest lecturer today as the part of the 3-in-1 program for the fertilizer and fertilization technology. So this 3-in-1 program aim to strengthen the student, to strengthen the knowledge of the student in globally, and we thought that the greenhouse gases emission in agricultural system, is one of the factor that affecting the greenhouse gas emission in agriculture system is from the soil management, particularly fertilizer application. And then that's why this topic is very interested, for our student, because there's no research in our department yet or in our faculty related with the greenhouse gas emission, especially from fertilizer application. Until now, there's still I just maybe many just a little bit research conducted for that. So that's why we invite Dr. Anna Majid, because I know Dr. Anna Majid is an expert in the greenhouse gas emission, and several or a lot of publication in the high or in the good journal have been published by Dr. Anna Majid. Before we starting the course, I would like to inform to all the students and also all the participants or lecturer who join the course today that first we will I will explain the curriculum vitae of Dr. Anna Majid, and then it will be continued by presentation from Dr. Anna Majid, and then discussion with the student. So I hope all students following this course seriously, and has or ask a lot of in the discussion, because I think this course is very interesting. Okay, so first I would like to inform or explain the curriculum vitae of Dr. Anna Majid. Please, the organizer Nova to see the curriculum vitae of Dr. Anna Majid. So Dr. Anna Majid is the assistant professor in the George August Universitat of Göttingen. She's expertise in environmental science. Her experience in teaching in the practical course in agronomy, and then crop production and grassland management. And then the last is application of explorative data analysis to agronomy from 2020. And then there's many selected publication from Dr. Anna Majid. The first is Measure Greenhouse Gas Budget Challenge Emission Saving from Palm Oil Biodiesel. I think this just published in this year, and it's in a good journal, and then on 2018, that's Dr. Anna published the article entitled Impact of Forest Conversion to Oil Palm and Rubber Plantation on Microclimate and the role of the 2015 ENSO event. I think you can see the curriculum vitae of Dr. Anna Majid. So as I know that when I conducted PhD in the Göttingen, Dr. Anna Majid is a postdoctoral student, or he worked in the postdoctoral program in the same project in the CRC 90 in Jambi, Sumatra. Okay, I think that's all for the curriculum vitae in of Dr. Anna Majid. Now we start the course of the International Guest Lecture by Dr. Anna Majid. Dr. Anna Majid, time is yours.
Okay. First of all, thanks, Sharul, for the invitation. I'm very pleased to be here. Selamat sore. I have nearly forgotten all my Bahasa, so I cannot help much there. But I am very happy to be here and to see that so many people are interested in this lecture. And I think this can open many opportunities for collaboration, and this is very, very interesting. As Dr. Sharul mentioned, we've worked together for several years in a project when I was working in Indonesia. And we got to know each other and to work very well. So I hope this is a good opportunity to open new collaborations and you find this interesting. So I'm gonna now share my screen on a presentation that I've prepared on the topic, and I think you see my presentation. I think this should be okay now. Yeah. If there is any problem with the presentation, please let me know, okay?
Yes, yes. Okay, we will inform you, Anna.
Yeah, because these things happen in the digital lecturing, that even if you have prepared everything, sometimes things do not work. So as Sharul mentioned in this very nice introduction, I'm working in greenhouse gases from different agricultural systems. At the moment, I'm an assistant professor in the Department of Crop Science in the Division Agronomy from the University of Göttingen. So I'm going to present you a bit on what I work, and we are going to talk a bit in general about agriculture and greenhouse gas emissions. Then we will talk about the different mechanisms that lead to production of greenhouse gases and how they are related to fertilization, because I know this is a fertilization program. So I wanted to just stay a little bit in the topic. Then we are going to talk about measurement techniques. So how do we measure greenhouse gases? Very briefly, just so that you know how we lead or have the results that I'm going to present. And then finally, I will present some case studies. And these are some research that I've been doing during my career in different agricultural systems, mainly in Europe and in Indonesia. I will present some results from Indonesia as well, just the last one, because I think this might lead to some discussion, and I hope you find this interesting. But also a couple of studies that I am not co-authored, just that I thought would be interesting to put the global picture of greenhouse gases appropriately. So we start with agriculture and greenhouse gas emissions, and first, I wanted to mention how ammonia synthesis has changed the world. So this is a paper from 2008, which shows the increase of fertilizer and how this led to increase of meat production and increase of the world population. And these authors claim that the production of synthetic fertilizer, nitrogen fertilizer has allowed for the increases of the population. Without fertilizer, we wouldn't have the population as high as we have, because this is what allows us to feed the big population that we have in the world. So we can see in this slide another different paper where we see an increase in the upper figure, the increase in worldwide cereal production. The next figure, how it's similar, this is from 1960 to the year 2000, increase in nitrogen and phosphorus fertilizer use, and the increase in irrigation. But also something a bit more negative, the increase, or well, with some other problems, the increase in pesticide use. So, of course, with increased pesticides and increased nitrogen and phosphorus fertilizer and increased irrigation, the cereal production and the crop production has increased. And this is good because we have to feed big populations, but these have some consequences. So we see that the concentration of nitrous oxide, one of the most important greenhouse gases, because it's a very strong greenhouse gas, has been increasing since 1860. Here is a figure up to the year 2000. But it also mentions that the synthetic fertilizer alone cannot explain the increases of N2O. So this shows that the increases of N2O emissions are caused very likely not only because of the mineral fertilizer, but also because of the use of organic fertilizer. So related to that, we see this IPCC, so the Intergovernmental Panel on Climate Change, that shows that greenhouse gases have been increasing more or less since the Industrial Revolution, and the CO2 increased, methane, N2O, and the concentration of these gases in the atmosphere continues getting higher. Here we have data from last year from the World Meteorological Organization, who is the organism that measures the concentrations of the different gases in the atmosphere. And they saw that from the year 1750 until 2019, there was an increase of greenhouse gases, as you can see in the table, of 147% for the CO2, 259% for the methane, and 123% for the N2O. And we still see relative increases for the year 2017 to 2018. So this has been increasing and continues increasing every year. And we can see here what are the causes of these greenhouse gas emissions. And we can see that N2O is 6.2% of the total anthropogenic emissions, methane 16%, and CO2 13%. CO2, which we are talking about, this is a category that is called FOLU, which is called forestry and other land uses, where agriculture is included. So these are the reporting of the IPCC for the last period. And here we see that how is this share of the different components that lead to the greenhouse gas emissions, and how agriculture is responsible for 10 to 12% of the man-made greenhouse gases. And this is one of the largest sectors contributing to non-CO2 greenhouse gases. So we see that greenhouse gases are increasing. Fertilizer use is one of the reasons, and agriculture is one of the responsibles of this increase of greenhouse gases. So we see that N2O is one of the most important greenhouse gases in agriculture. We see in this figure, in the lower part, these red bars are land use change and forestry. The next one is drain peat and peat fires. So this is something very relevant for Indonesia, as you have a lot of peat and peat fires. The next orange section is enteric fermentation. So this is the cows that lead to methane. Then is rice cultivation, also relevant for Indonesia. And we are going to talk about some studies on rice as well today. Manure management, and then come the synthetic fertilizers, crop residues. So all the upper part is manure and applied to soil, manure applied to pasture, synthetic fertilizers, etc. So N2O is a really relevant greenhouse gas, even if there's also methane and CO2. And for N2O, agricultural soils are the main source of the N2O emissions, even if there's also a manure management or human sewage. But the most important is N2O. It's, agricultural soils. So we know now that agricultural soils are important for greenhouse gas emissions, and we are going to talk about which mechanisms are responsible for these emissions and how are they produced. So if we want to be able to quantify the emissions and be able to reduce these emissions, we have to understand which are the mechanisms that lead to these emissions. So N2O, as we said, is one of the most potent or more strong greenhouse gases, and it can be produced both in anaerobic conditions, when ammonia is transformed into nitrate through the process of nitrification, and N2O is produced as a byproduct. But then this nitrate is, in anaerobic conditions, transformed into N2 through the process of denitrification, and then this also leads to N2O formation. There's an additional pathway, which is nitrifier denitrification, which is produced by denitrifiers under anaerobic conditions, but this is in proportion, usually not that much. The share of emissions is much smaller. So then these three pathways are the main mechanisms responsible for producing N2O, and this shows that applying fertilizers which lead to increases in ammonia in the soil or ammonium in the soil and nitrate have the potential to increase N2O emissions. Also, emissions are influenced by the amount of water in the soil and by carbon contents in the soil and temperature. So here we see a figure of a paper where we can see an N2O peak. So we see that the first days the emissions are higher and then they get lower. So this first peak is caused by the application of fertilizer. So this is regular measurements in the soil with chambers. I will explain later how this is done and we see how the emissions increase afterwards. We talk now about methane. So methane in the soil, there's two mechanisms happening in parallel. There can be production of methane by methanogenic microorganisms. This happens under anaerobic conditions, so usually in waterlogged soils. So there's where rice paddy fields are very important because they are usually under water conditions. And then there is consumption of methane when there is air in the soil. And this is done by an enzyme, the methane monoxygenase, that is responsible of methane transformation in the soil. So again, these mechanisms are affected by the amount of water in the soil, which is responsible of the amount of oxygen in the soil, mineral nitrogen, soil organic carbon, and temperature. And mineral nitrogen, we are going to see now how, because this influences the relationships between a production of methane and fertilization. And that's why I wanted to bring this up here. So at the plant ecosystem level, if we fertilize with nitrogen, the plant increases and provides then organic carbon in the soil, which favors the methane producer. So this leads to increases on methane production. Also, the nitrogen favors the development of the methane oxidizers, so it also favors methane consumption. But at the biochemical level, there is an interaction with the enzyme that is responsible for the conversion of methane in the methanotrophy, where that it's inhibited by the ammonia. So if we apply fertilizer, this methane cannot consume this enzyme cannot consume methane. So these are the relationships with fertilizer, and that's why emissions of methane are different in fertilized soils than in not fertilized soils. Then we have CO2, the third gas, and I want to briefly show which main fluxes of CO2 we are going to be talking about. So there is soil respiration, which can be autotrophic by the roots, and heterotrophic from which is the microbial respiration, anaerobic and anaerobic. We are going to talk possibly about ecosystem respiration, which is soil respiration plus plant respiration. And then net ecosystem exchange, which is the difference between photosynthesis and ecosystem respiration. Here, the results I'm going to be presenting are going to be mainly about soil respiration when we measure with chambers in the soil, and net ecosystem exchange in 0.3 when we are measuring at the ecosystem level. So we are going to see here which measurement techniques are the most common for measuring greenhouse gases, and which ones I have been using and which can be useful maybe also for you in the future if you would be interested in getting into this topic. So for measuring greenhouse gases, it depends on the scale you are working at. So you can measure at the leaf level. Then there are some instruments which are called IRGAs, infrared gas analyzers, that measure at the leaf level and measure the exchange of CO2 and water in the leaf. You can measure at the plant and soil level, and then usually you use chambers, and there's different type of chambers which are big and small, and we will talk about them. We can also measure at the ecosystem level using techniques like Edicovariance, and this is a technique I've been using in Indonesia, in Jambi, and I will show you some results about our measurements with Edicovariance, but also with chambers. So these two techniques in the middle are the scales we are going to be working with today, but there's also measurements at regional level, which are done with modeling or with remote sensing. So satellite images, also drones. There's several options on how you can measure at regional level. But today, this is more my field of expertise on lower scales or smaller scales, and we are going to talk today mainly about plant and soil and ecosystem level measurements. The measurement techniques can also be divided into depending on the principle that you use for measuring. So not only about the scale, how big or small the scale is, but the principle you use. In this case, we're going to distinguish between chamber measurements, which usually, as we said, measure at the plant soil level, micrometeorological techniques, which measure at the ecosystem level, then remote sensing and modeling. So remote sensing and modeling usually work at bigger scales, but modeling can be also done at smaller scales. So then this is why it's interesting to split between a modeling and remote sensing, not only as they are different techniques and can work in different scales. Okay, so these are the techniques. And today, as I said, we are going to be mainly talking about chamber measurements. And a chamber is a very simple, usually homemade or custom made when you use manual chambers, and that's what I'm going to be showing here. A system in which you prepare a chamber that you put into the soil, and then you collect gases with a syringe, and then they are stored in glass excitaners, and you measure the concentration of the gas at different times during the closure of the chamber. Here was every 10 minutes. And we analyze the data in a gas chromatograph, and we study the linear increase in concentration with time to estimate the flux. So this is quite a simple method if you have a system to measure the emissions, which you need a gas chromatograph, which is the most complicated part of the system, having the gas chromatograph. The other technique we're going to talk about today was Edicovariance. So this is measurements at big scale. And we can use it for measuring greenhouse gases or energy fluxes, water, heat. So, but we are going to use it for CO2 or water, N2O, methane. And this has the advantage of you can measure 10 to 20 times per second, and you average it over 30 minutes. So you put a tower in the field with your instruments on the top, as you can see here, and when the wind comes, you measure what the wind brings, and with this and how the increase of concentration go up and down, the molecules move, you can estimate the flux. So I think this is a bit more difficult to understand, and in the end, the idea is that the tower would be like as if would be in the center of a chamber, imaginary chamber, and you assume that the flux that goes in and goes out is identical, and so that what you measure, the differences are the vertical exchanges, and this is what we measure here. So this is the technique. Some of the results I'm going to present are based on. And these are also the two most common techniques for measuring greenhouse gases. Chambers are usually cheaper and easier to manipulate if you have a gas chromatograph, but you have to measure manually, and how much information you get depends on how many samples you take. The more you sample, the more information you have. But it has the advantage that you can do that everywhere because it's not complicated to transport instrumentation, etc. While for Edicovariance, you need electricity, and this is sometimes more complicated. So this is not possible to do it all the time. In Indonesia, there are several places where Edicovariance fluxes are being measured, not only in Jambi, but also in Palu. And I think there were some other towers in northern Sumatra, but this is just a bit more limited compared to the possibilities of chamber measurements, also because of how expensive they are. But yeah, the idea is that we measure the vertical fluxes because flux in and flux out is identical. And then it's called Edicovariance because we calculate the covariance between the different components. And it can be used for different gases, water, CO2, methane, N2O, etc. If you want more information, all the presentations have links that you can access the papers. So yeah, here the sensors to measure the concentration of the gas, in this case is CO2, and an anemometer to measure wind. So now that we've made a bit of a bigger of an introduction on the bigger picture on why is agriculture responsible of emissions, and why are emissions important, and how do we measure them, I'm going to present studies on different systems, mainly agricultural systems, but I think one of the studies is not only an agricultural system. I try to present mainly studies where I have participated and I have been involved during my career. I have worked mainly in Spain, Italy, and Indonesia. So these are the locations and now in Germany. So these are the locations where I have measured and from where I have some data. But also I brought up some other studies that I thought could be very interesting and are good to get a bigger picture of greenhouse gases. So this is one of the studies of my PhD, where we were measuring different peak slurries with different treatments. Some were fresh peak slurry, and when some were composted, some were anaerobically digested, and we applied them to a maize crop. And we see in this figure, what I want to see is how when you apply the fertilizers, and this is indicated by the vertical arrow at the beginning of each graph, there is an increase in ammonia and nitrate in the soil. So these are the concentrations of soil ammonia and nitrate due to the application of fertilizers. And depending on the different fertilizers, these increases are faster or higher, lower, and they remain high for longer or shorter periods of time. So then in this same study, we see what happens with N2O emissions. And this was measured with chambers. And as you see, this was quite a lot of replicates. And there's the emissions happen at the beginning are very low, and after the fertilizer application, they start increasing. And they are different depending on the composition of the fertilizer. So different fertilizers lead to different emissions, and organic and mineral fertilizers result in different emissions. One important thing is that this is different depending on the soil and climatic conditions. So the results we obtain for a Mediterranean climate like in Spain might be different to the ones we obtain in a tropical place like Indonesia. But of course, you are still going to find differences between the different fertilizers, just that these might not be exactly the same. Here, it's another study on an olive grove, so a plantation of olives to make olive oil, which is where we are studying two different management practices. So when I first started working in an olive plantation, it reminded me a little bit about an oil palm plantation because it's the same idea of a monoculture, and many people also have removed all the vegetation on the ground. So then the soils remain bare in many places, and this has big problems on soil erosion and losses of carbon in the soil and water runoff, etc. So then there is an alternative management practice, which is to keep the weed on the ground. But this is an ecosystem which is very water limited. So it's very different to the Indonesian oil palm plantations, palm oil plantations where there is plenty of water because rainfall there is very high. And then we wanted to see what are the effects on the fluxes. So this was measured with Edicovariance, and that's, we can go back, one of the towers we had in the sites. They were two identical towers in two different sites. And what we see here is that the CO2 uptake was higher when there was a weed cover. So the ecosystem was capturing more CO2 in the presence of weed cover. So I also tried to study what happens with other greenhouse gases, what happens to N2O and methane. Is there a difference? And what we see on the left side is with the N2O, and we see that there were no clear differences in N2O emissions. These were measurements for a full year, once a month. And on the right side, we see methane, and it feels that there is a little bit higher methane uptake in the weed-free treatment, so where there is no weed kept. Because in the end, you have to look at the effect of the three greenhouse gases and not just one by its own. So this is another study I was involved in, and this is in Italy. It's a rice paddy field, and rice paddy fields are relevant because, well, first of all, because rice is one of the most relevant crops worldwide. In Europe, there is very, very little rice production. It's mainly concentrated in Italy and Spain. Those are the two bigger points. But certainly in Asia, this is a big thing. And there is a lot of studies on rice in Asia. But this was one of the first where we studied methane emissions in a rice paddy field with Edicovariance. So this technology became available and we measured soon after. But you can see that there's also some pictures of chambers. So we were using both techniques in parallel. And this was very interesting because it allows to compare what are the benefits of one and the other. So the chambers give you information about what happens in different spots in the soil. The Edicovariance gives the high temporal resolution and has an homogeneous measurement over the whole field. So this is very interesting, and you can compare what are the estimates you get from both techniques. So this is very, very useful to have the opportunity to measure with both techniques. And what we saw here, you can see in the upper figure that the black dots are the measurements with chambers, and the gray circles that look like dots, but they are circles, are with Edicovariance. So the fluxes do follow the same pattern. But in the end, we saw a little bit lower emissions with Edicovariance. But the interesting result here is that we measured for 2 consecutive years, and the emissions of the second year were much lower. And this was because in the middle of the growing season, there was a drainage of the water table. So the water table was removed, it was lowered, and then it was put back again. This removes so it lowers the emissions. Depending on where you are, the water table is there because the rice grows very well there, and other weeds do not grow well there. So that's why they are usually kept there. But it seems that if it's removed for a short period of time, it's not enough for the weeds to develop, and you can still not have a big impact on yield, but have a benefit on the methane emissions. And the figure a bit down, it's a relationship between the water table depth and the average methane fluxes. So the higher the water table depth, the higher are the mean daily emissions. So if you want to keep water in the rice paddy field, you can keep it, but maybe with lower quantity would be enough, and then the emissions which would be much lower. We also measure N2O. As we had these chamber measurements, we use them to measure N2O, and we saw that they are related to fertilizer application, and when you remove the water, then the N2O emissions can increase. So then you have to make sure that the increases of N2O are not counterbalancing the benefit of decreasing methane. So what we did here is analyzing the whole effect on greenhouse gases of the 3 gases, methane, N2O, and CO2. And if you're a farmer, you might be very aware of the environmental problems and you might care about greenhouse gases, but you are very interested about what your yields are. So farmers are not willing to implement management practices that lead to decreases in crop yield. So in the end, there is a term which is called greenhouse gas intensity, which relates the greenhouse gas emissions per yield, per gram of crop yield. So I think this is important to see that when in the second year, 2010, when we removed the water table, the greenhouse gas emissions per crop yield so the yield was lower, but also the emissions per crop yield. In Europe, there's discussions about if farmers implement these sort of techniques, there should be a way to compensate their loss of yield. But yeah, this is complicated, and that's why farmers prefer to keep the most efficient management practices, even if they are not the best regarding greenhouse gases. So this is another study. This time is something I was not involved in, but these are emissions that are not relevant in tropical systems, but are relevant in temperate regions, which is the fact that the soil freezes in winter and then defreezes a few days later and freezes again, and these are called freeze-thaw cycles. And these freeze-thaw cycles increase N2O emissions, and these are relevant and the emissions are related to the number of freezing days. So if you ignore these freeze-thaw cycles, which sometimes happens because traditionally people were saying, okay, it's winter, the soil is frozen, we don't have to measure. Well, these emissions do have a contribution to the annual budgets and can lead to big underestimations. This is another study on nitrification inhibitors. So I've used nitrification inhibitors in Mediterranean cropping systems, both in maize and in barley, and we saw decreases on emissions. We can see here that there's decreases of there's here two different nitrification inhibitors, the DMPP and the DCD, and both lead to 38 and 35% reduction on the emissions. Okay, so, and this is another study, which is also not one where I have been involved, but I thought it would be interesting to talk about organic farming, because this is becoming more and more relevant to talk about organic farming, but it's important to see what are the effects on greenhouse gases as well. So there was a decrease on N2O emissions in the organic farming. The yield scale cumulative N2O emissions, so really similar to the greenhouse gas intensity, were similar between organic and non-organic. So this shows that organic farming can be a system to contribute to greenhouse gas mitigation. So here they have different farming systems that they tested, and they saw that decrease on N2O emissions. And this is a reed wetland. So this is not an agricultural system, but this is another study where I was recently working, where we had the opportunity to measure with Edicovariance. Here we didn't measure with chambers for many consecutive years. So here we have 6 years of data, up to 6.5, nearly 7. And we measure CO2 for several years, and then methane for 3.5 years, which starts giving information not only about what happens one single year when you measure, but what happens with the temporal variability of the fluxes. So this is, and you see that there is large differences in the fluxes. Also in the middle, you see evapotranspiration. This is the water flux. And there is a large variability from one year to another, also in methane fluxes. So what we found out is that the large temporal variability was related to a large seasonal variability. So this reed is grows in spring and then dries up in summer, and then has a senescence period, and then during this, there's a transition and then a senescence period. So then during these different periods, the emissions are very different. So both also the fluxes of carbon, methane, and the water fluxes are very different in these different periods. So then we see that there was, when we look at the accumulated fluxes of CO2 and methane, we see large variability. It can be that 1 year the system behaves as a sink, and the next, and a very strong sink, and the next year is a source. So there is a big functional vulnerability to climate variability in this type of Mediterranean wetlands. So as you see, I have a lot of examples from Mediterranean systems because I worked quite a lot there, but also because these are very sensitive ecosystems and that suffer from droughts and have very high temperatures. So yeah, this is why some of these studies are here presented. And finally, I want to show my results about oil palm plantations. And I guess many of you might know that both Dr. Sharul Kurniawan and I were involved in a big research project from the University of Göttingen with several Indonesian universities, where we were measuring, studying different land uses, oil palm, rubber, forest, jungle rubber in Indonesia, in Jambi. And my job was mainly to measure greenhouse gas emissions. So what we did is, using this Edicovariance measurement technique that I have shown you, measure in a young plantation, which is a plantation that is not yet productive, which means that it is not producing yields, not producing yield yet, no fruits. So this was a 1-year-old plantation. You can see the picture on the left. And then in a mature productive plantation, this was a 12-year-old plantation. And what we saw is that while the young plantation was a source of carbon with very high carbon emissions, the mature plantation was a strong carbon sink. So we think that the young plantation is a very high source because this might have been transformed from forest, and we are still seeing some of the soil emissions and some of the carbon losses from the soil, and the palms are too small so that the photosynthesis they do cannot compensate these high losses. But then later, when the plantations become mature and the palms grow a lot and they are very big, then the emissions, then they do a lot of photosynthesis, and then the emissions are turn into negative. So here it's a full...
The negative mean, what's the negative? The meaning of negative is decrease?
Yeah, I can go back. Thanks for the question. Yeah, so negative means, sorry, I didn't clarify, so you can see these arrows in the figures. Negative means that the ecosystem is absorbing carbon from the atmosphere, while positive means that the ecosystem is releasing carbon to the atmosphere. So this shows that young plantations are not very good for the climate because they are releasing carbon to the atmosphere, while mature plantations are positive or can have a benefit because they are capturing carbon from the atmosphere. So then when you look at a plantation, you shouldn't look at 2 single points, but you should look at a full life cycle of the plantation, and we try to make estimates of the full life cycle. But here, first, I want to show the results of the first-year-old 1-year-old plantation and 12-year-old. So both on mineral soils, this is also important. They both were measured in mineral soils. And we see that in the 1-year-old plantation, there is no yield. Here, there was a yield of 900 plus minus 49 grams of carbon per square meter per year. We see that this is the same number as we saw before, NEE. So the net ecosystem exchange, the exchange of gases, which is the balance between the photosynthesis that the ecosystem does and what the plants and the soil is respiring is positive in the young plantation and it's negative in the mature plantation. What we see is that methane emissions are the same because it's not much affected. So we are estimating that they are similar. And N2O emissions are much higher in the mature plantation. And this is something that is related. So as we said, N2O emission is very related to fertilization rates. And I think this might have been a particular case on our study because the young plantation was a smallholder plantation, which applies much smaller fertilization rates than the industrial plantation. That the industrial plantations, they apply much larger amounts of nitrogen fertilizer. So then the emissions are much higher. But from what we understood in general, young plantations do not apply so much fertilizer. Also, it seems to be common to grow different crops between the different rows. In young plantations, this doesn't happen, and this might also take some of the nitrogen and reduce the emissions, and this doesn't happen in mature plantations. So large differences at different stages of development, and when we want to analyze what the effects of oil palm cultivation are, we should look at the whole life cycle and not just what happens when they are mature, because the picture is very positive regarding climate. It captures a lot of carbon, not only when they are small, because the picture is very negative. It's important to look at both. But another important thing is that depending on the soil type, the emissions are very different. So here we compare in the 12-year-old plantation mineral soils versus peat soils. And this, I want to clarify that it was not a full plantation on peat soils. It was just some areas of the plantations that were peat soils and where we decided to do measurements. And then the estimates that we have at the ecosystem levels are not our real measurements, but measurements from other locations in Malaysia or in other plantations in peat. But we do have measurements of carbon and nitrogen in the soil to show that the soils were very different there. And we measured N2O and methane with chambers in these mineral soils and peat soils. And for the methane, we see that it's much larger in the peat, and also the N2O emissions are much, much larger in the peat. So, and then the net ecosystem exchange, again, it's much smaller. So I think there's a large potential for greenhouse gas emissions on plantations in peat soils, and this is something we should consider. It seems that from Europe we always want to show the bad part of oil palms and this on oil palm plantations, and this is not the case. I think the idea of this study is to show that oil palm plantations should not be placed on peat soils, and that is much better if they are done on mineral soils. And that's why I'm going to first explain this slide, and I'm going to then show you my final slide where I want to bring the positive picture on, so that you don't end up with the idea that we think that we only bring up the problems, but also we want to bring up some solutions. So as I was saying, we shouldn't only look at the young plantation or at the old plantation, but we have to look at the whole life cycle of the plantations. And when you look at, and here we look at the whole life cycle, not only of the plantation stage, but also what happens in the whole production of biodiesel. So this means that the fruits are harvested in the plantation and then taken to a mill, and then the oil is pressed, and then this oil is taken to a place where you produce biodiesel. And then this has different sort of emissions in different stages, and we try to estimate it. And usually for biofuels, it's assumed carbon neutrality. Whatever is captured is lost, so nobody makes a balance for CO2. And what we saw here is because the ecosystem emissions in the young plantation are so high due to the previous deforestation, then the emissions are very high. And this shows that there is land use change emissions from forest conversion, in the bottom part, but also by emissions of CO2 in different stages once you burn the biodiesel, but also in different stages of the production. And that the sinks, the CO2 capture cannot compensate for that. So here, this is where we want to bring the positive picture into this story, because first we're going to look at the figure on the left side, and it's a biodiesel from oil palm plantations in first rotation cycle after forest conversion. So we found out that a forest conversion has really large emissions, and then plantations, when they are very young, have very large emissions. So the way to reduce the greenhouse gas emissions from oil palm plantations or from oil palm biodiesel would be either to increase the rotation cycles. So usually in Indonesia, this is done for 25 years. But what we heard is that this is in some places done up to 30 years. I think in Africa, in some places, they started to do up to 40 years. I think this is quite uncommon in Indonesia, but we just wanted to make the theoretical study and also aim at early yielding varieties so that we are lower time or less time on the phase of low emission, of very high emissions of young plantations. You can see here that there is a line in the middle of the graph that says reference fossil fuel emissions. And this is reference is a number that the European Union has set on the emissions, that the maximum emissions that a biofuel can have to be called biofuel in the European Union. So what we wanted to check is what happens with the emission, with the biofuel, with the biodiesel from palm oil. Does it comply with the regulations from the European Union? And what different strategies can we do to make it comply with the requirements so that this biodiesel is according to the requirements of the European Union? So then the first analysis is a traditional LCA, where we are assuming this carbon neutrality that I was mentioning before, and this is something where the emissions are a bit lower than we're going to get later, but this is also not complying. So every point that is above this reference fossil fuel emissions line shows that these emissions are too high for the requirements of the European Union. So we have this business as usual, which is our analysis where we have incorporated our measurements in the life cycle analysis. And then we have the different scenarios with different rotation cycles a bit longer, or scenario C has early yielding varieties. And we see that all these scenarios can lead to reductions of the emissions, but still their points, the black dots, are always above the line of reference fossil fuel emissions. However, on the right side, when we look at the right side, all the points are below this line. And what is the difference here? Well, the difference here is that we are assuming that this biodiesel is coming from plantations which are not in the first rotation cycle, but after forest conversion, but the second rotation cycle. So this is a second rotation cycle on a previous plantation, or that they are established on degraded lands. Because most of the emissions are happening due to the large emissions due to land use change and the sequestration that the forest would have done if it would have been there. If we manage to remove all these emissions, then we can have a biodiesel that complies with the regulations of the European Union. And then, of course, increasing the rotation cycles is a helpful option. So this is the message we want to give here, or I wanted to give here, that palm oil is an oil that is necessary worldwide because it's a very effective crop, but it should not be established on soils that were previously forests, because this leads to a lot of carbon losses. If they have to be established, they should be on places that are already deforested and are already plantations, or on degraded lands. And this has a large impact on the carbon budget. So with this figure on the different emissions of carbon and N2O, CO2 in different land use changes, I want to finish my presentation, and I am happy to take your questions.
Yeah, okay. Thank you very much, Dr. Anna, for the nice presentation. Yeah, that's I guess that's a nice research, nice topic. Yeah. Also, it's expensive, I think. Yeah, with the Edicovariance, as you say. Yeah.
Yeah, it is expensive. Yeah.
I don't know. Maybe it's a billion in rupiah, yeah, in Indonesia.
Yeah, it's very expensive, and it also needs a lot of expertise. That's why I wanted to present these chamber measurements, because I think these are the way to start at the beginning when you want to start developing and having information of ecosystems that you don't know. Usually chambers are a very good opportunity.
Yeah. Okay, thank you very much, Dr. Anna. And so if I open now the discussion session for the student, yeah, please write your question in the chat room, or if is it possible in the raise hand, Bu Nova, yeah. Okay, maybe first I would like to invite Hana Siva, yeah, Hana Siva, please turn on your microphone and camera, yeah.
Okay.
Hana Siva for class A, yeah.
Is it my voice clear for you? Clear? Okay. Okay,
Yeah.
thank you. I want to ask about our fertilizer can make gas emission, but we needed to plant. So is there any material that can replace for plant like so like what I say so with for replace fertilizer, because fertilizer is used for nutrition for plant. Is there any material can replace it? Thank you.
Okay, thank you, Hana. Yeah. Okay, Hana, you want to explain each question, or
Yeah, I can.
Okay.
Yeah, I think it's easier if we do one by one. Okay. So fertilizer increases emission, but of course it's necessary for the plant, as you say, certainly for growing. Of course, there's other materials like organic amendments, like slurries from different animals, from cows, or from waste. You can also do compost from crop residues, or from different residues that can be used also to fertilize your soil. And this is not a matter of not using fertilizer, it's a matter of using enough amount of fertilizer, but not too much, so that there is not too much fertilizer that ends up leaching into the soil. And this Dr. Sharul is much more expert than I am on nitrate leaching. He's been working a lot on that. So it's just the excess of nutrients what ends up in higher emissions and higher nitrate leaching.
Okay, how's Hannah? Is answer your question? Is it? Hannah? Is it?
Okay, thank you. It can answer my question. Thank you for your answer.
Okay, thank you, Hannah. Then we continue for the next student, Ahmad Jantang Winayapana. Okay, please, Ahmad, you can turn on your microphone and camera.
Thank you, Mr. Sahul, for the opportunity. So, what I want to ask to Dr. Anna is there any tool or method for estimating, quantifying, or calculating the emission of greenhouse emission GHG in wastewater treatment plant? Thank you.
So if I understood correct, you want to know if there's techniques to measure emissions in wastewater, right?
Yes.
Yeah. So there are, you can, I'm really not an expert on that at all. So, but there are, and there's people estimating these emissions on wastewater. And it depends where. So for example, I know that in the oil palm mills, the residual waters are just left in lagoons to ferment, and this leads to lots of CO2 and methane emissions. If you are measuring on a lagoon, you could use floating chambers. So this also works like in the soil, but they are floating. You can also you put them on the surface, like, and then you wait for the gases to accumulate. But there's also techniques on how much dissolved methane, for example, of dissolved CO2 is in the water, and with that you can estimate the emissions. Yeah, there's different options and depends on where your wastewater is. And wastewater management plants, when they are accumulating the wastewater, usually try to comply this or measure some of these emissions.
Yeah.
I hope this helped.
Yeah. Okay, Ahmad. Yeah. I think it's answer your question. Yeah. Thank you, Ahmad.
Thank you,
Now,
Mr. Sahil and Dr.
yeah, thank you. Now we continue for the next student is Gabriel Esther, yeah, from the class E. Okay, I think the class E is the English class, Anna.
Okay.
Yeah.
Yeah, okay. Is my voice is clear? Is my voice clear?
Yep.
Yeah, so I want to ask about the gas emissions management, where the gas emissions itself in Indonesia is one of the major problems of agricultural sector, where Indonesia is known as the lungs of the world. So we need to manage this in proper manner. So I have read several articles that soil repairs such as biochar itself can reduce gas emissions or greenhouse emissions. Based on your point of view, the biochar is effective enough to be used in reducing greenhouse gas emission in the agricultural sector. And the second question is, do you have other methods that can be used more effective, easier, and environmental friendly to reducing these greenhouse gas emission? Because in our country, one of the major problem is the farmer is really lazy to do complicated method to manage these problems. Thank you.
Okay. Okay, so thanks for your question. So you are asking on how we can reduce these emissions, right, in general, and what are the strategies to reduce emissions in agricultural soils. So the problem is that the management solutions are very specific to the different soils and ecosystems. So as I was telling you, for example, using organic fertilizers in Mediterranean systems is a way to reduce the emissions, but this is not necessarily the case in a different type of climates. So you need to do studies to understand what is your best way, and there's things, of course, these nitrification inhibitors would help for N2O emissions, but they are costly, and it's not only in Indonesia, everywhere the farmers want to get a profit and they don't want to invest more. So this is really not special for Indonesia. Everybody wants to get a profit, and not everybody is aware of environmental problems. Some are, and some are interested, but not all. And this is just something that happens. But I think one thing that could be interesting is that sometimes the fertilization levels are very, very high and are more than the crop really needs. And it is not even considered what the soil already has. So places like you are working at university where you can measure how much nitrogen is in the soil, so you know already how much there is and you don't have to apply that much. So I think just applying lower amounts of nitrogen in some places might not have an effect on the crops and might reduce the emissions. But also, I am not an expert on farming systems in Indonesia, and I think most likely some of your lecturers and professors in your faculty are much more experienced in this topic and would be able to tell you more about management practices there.
Yeah, thank you, Anna, for the it's a good answer. Yeah, and then I think Gabriel, it's answer your question. Yeah, from Dr. Anna. Yeah, and then Anna, there's still a lot of students want to ask because this is an interesting topic. Yeah.
Okay, yeah, sure, no problem.
Yeah. Okay, we continue to the next student is Jopi Maiski. Yeah, please.
Yeah, so my name is Jupi Miska. I would like to ask, are there any obstacles that you have found during you do the research about these greenhouse gas emissions? Thank you.
Okay, so it depends on where the obstacles you find are different. So, for example, in Indonesia, it was a challenge to find a place where we could measure that we would have the requirements we need in terms of electricity, for example. And this, and also that we found a plantation that would let us measure, and then whatever results we get, that we could publish. But then the challenges are different when you are in Europe, because maybe it's too cold and then the instruments freeze. So it's just these challenges are different depending on the locations that you have. I don't know if you mean these kind of challenges or you mean more social challenges. I hope this.
Yobi, please.
Does this explain your question?
Yes, thank you.
Okay, thank you, Yopi. Yeah, now I would like to choose from the other class because there's many from the class E. Yeah, now is from the class M. Anggita Skarhayu, please, Anggita.
Thank you for the opportunity. I want to ask, do farmers in the countries you have studied, or at least in German, have awareness of greenhouse gas emission? And if no, do you try to explain about the greenhouse gas emission production from their land, or at least explain how to reduce it to the farmers? Thank you.
Thanks. This is a good question. So I think it really depends on per country and per region in the country. So when we were working in Italy in the rice field, this was supposed to be an organic farming field, but they didn't fully comply with the requirements of organic farming. So they were not super aware of that. I think in Germany there's more awareness, but I think not only about greenhouse gases, which as well, but also in general about organic products that have less use of pesticides, for example, or herbicides. But this is mainly because the population wants a product that is organic. So then the farmers have to produce this type of product. So I think it's important to raise awareness in the farmers, but also in the general public, because if the general public wants products with a certain quality, in the end the farmers will have to adapt. But I think at the moment, like, it's very good that in Indonesia there starts to be people caring about these problems, because I think sooner or later this would make a difference. The largest emissions in Indonesia anyways are due to the forest conversion. So if you would raise awareness on not converting the forest into agricultural land, but using lands that are already converted, this would already make a huge impact into the emissions.
Okay, Anggita, is it enough? Yeah, I think it's already answered your question. Okay, because yeah, we also limited by the time. Yeah, I will well, maybe just two student next. Yeah, more. Yeah, Anna. Yeah, is it okay still?
Yeah, it's okay.
Yeah, okay. First, Sakila Luki Rainata, please turn on your microphone and camera, and then you can mention your question.
Yes. Hello, Miss Anna, and my name is Shakila. I'd like to ask, so from your presentation before, you have mentioned that one of the largest rice field in the Italy was in the Po Valley, and how they managed to minimize the CH4 there is by removing the water. And so I want to ask, how do you remove the water from the rice paddy field? Do you use some kind of tools or machines, or you depend on the climate change that happened there, or by the seasons that develop there? Because in Indonesia, we can just wait for dry season, so the water in the rice paddy field will get dry by itself. But how about there in Europe? Thank you.
So this is a very good question. So in Europe, in Italy, this is done automatically. So you cannot wait for the season because usually in the middle of the cropping season. So there's a first one difference. In Indonesia, sometimes you grow 2 or 3 rice per year in the same field, while in Europe we can only do one because then in winter it's too cold. So it has to be only in summer or spring in summer. So this is different. And then because the fields are labeled and then they are flooded, so there's water put there artificially, and then the water can be taken out with a little pump or with pumps, and then you take it out. And so, or because it continues evaporating, you don't add water for some days, and then it lowers the water table. Sometimes it's not necessary to fully dry it, but just lower it a little bit for 1 day or 2. But this is done not waiting for the climate, but just with human intervention. I hope this helps.
Yeah, thank you, Anna. Yeah, I think Shakila, it's already answered your question. Yeah, and now I think that's the last from the last student. Yeah, because we limited by the time. Yeah, I'm sorry if not all the students who raise hand can mention and ask the question to Dr. Anna. And then the last from Patricia Josephine from the class N. This because we have a lot of class Anna.
I see.
Yeah, we have like 500 students in the fertilizer and fertilization technology the course. Yeah.
Okay.
Okay.
Yeah. Okay.
Patricia.
Thank you for the opportunity. Hallo, guten Tag, Frau Anna.
Hello.
Wie geht's?
Good. My German is not very good. I'm not German. I'm Spanish. So
I see.
I think your German might be better than mine.
We are learning though. So first of all, I'm really thankful that you have explained about your research in Indonesia. It's really interesting. And my question is, why do you choose to put the Tower of Easy in oil palm plantations in Jambi? Is there or has there any prior research before? And how do you take the permission into the locals? Thank you.
Okay, so it was decided, well, before I got involved, no, but Jambi is one of the places where land use change is happening very fast, and it has happened in the last 20, 30 years very fast, and that's why this location was selected. And oil palm expansion is one of the main causes of forest conversion. So this was a big deal. And also at the worldwide level, there is a lot of controversy on what's happening with oil palm. So we wanted to have real measurements on what is happening there. Not, because some of the information that was out there was not based, at least there was no full ecosystem measurements with Edicovariance. So this is why we decided to put it in Jambi. And then the plantations we found, well, that was quite of a challenge until we found plantations where they allowed us to measure. So this is, but yeah, we measured, and I think you had a second question on why in Jambi, and what was the second thing? Like oil palm and Jambi, and you had an additional question.
Yeah, maybe it's yeah. I will ask that's related with the permission because the research is collaborate with the local university. Yeah.
Yeah, sure. And then exactly, well, we had permissions through the local university, but you also ask how do we inform the locals about our results. And I have to say, I haven't done much. I have done a few lectures like I'm doing with you today, explaining our results. But I think the project we were working with, this CRC 990, has now a new component which is on education, and they are trying to take the results from research to make it to high school institutes and bachelor, like universities, to have material to teach information about the outcomes of the project. So that, because I think the scientific results are very important, but it's also important that this goes to the general population, and especially the population of the place where this is happening. So I think this is starting to happen at the moment. And I think if you go to the website of the CRC 990, you can see that there is now an educational component, something like that, and you can find more information. If not, I'm sure Sharul can help you find, or I can do as well.
Yeah. Okay. Thank you very much, Anna, for the answer. Yeah. For the all student, I really sorry that I can give you the opportunity to ask to the Dr. Anna, because I think now we have to stop the course, yeah, because it's already more than an hour, yeah, almost 1.5 hours. Yeah, I think in Göttingen that's 11:30. Yeah, almost 11:30. Yeah, but if I can summarize, yeah, from the presentation of Dr. Anna, that's we can learn that fertilization, yeah, lead to increase greenhouse gas emission, yeah, and then the increases of dose of nitrogen fertilization also impact to increase greenhouse gas emission, especially on nitrogen emission. As then in the paddy soil, that's also the water level impact to the greenhouse gas or in the methane emission, yeah, and also nitrogen fertilization in the paddy has an effect to the nitrogen emission. I think that's, and then there's many measurement technique. Yeah, in our department we have a GC, Anna. So
okay, nice.
We can discuss later after this course. Yeah, but the potential collaboration in the research in the future. Yeah, I think we have to stop in the course, yeah, for the course, yeah, because we will continue discussion with Anna, with the many our lecturer, yeah, because okay, before we stop this, Anna, is it possible if we get your presentation maybe in the PDF file, yeah?
Yeah, I will send it as PDF.
And also, is it possible if we set your presentation to enrich our teaching material for the student? Yeah, we will put your name in the slide, in the material. Yeah.
Yeah. Okay.
Yeah, I think that's all for the course today. We give applause to Dr. Anna. Thank you very much, Dr. Anna. Also, thank you very much to all students who join in the course today. Yeah, I saw that 300 participants in the Zoom. Yeah, when you presented your material, Anna. And also I think a lot of participants also join in the YouTube channel. Yeah, because this is a very interesting topic.
Thanks a lot for the invitation and for all the interest and all the questions. And yeah, it's a pleasure.
Yeah. Okay. Thank you, Anna. And so the student can leave the Zoom. Yeah, because we will continue with the discussion with Anna. Yeah. Thank you very much, Anna.
You're welcome.
Yeah. I think we need like around 15 or 20 minutes. Is it okay, Anna?
Yep. I still